Alright, so welcome again. And we're going to stick with fluorescence and keep investigating the subject. Fourth lecture of the second week. And this is a good time to credit the proprietor of the lecture slides that we're working with, Professor Attila. It's a good time to credit him for that. So thank you, Attila. And we're going to go through spectral distribution, what we need to know about spectral distribution, different parameters. I'm only going to go through the ones I saw represented in exams. I'm not going to put information that you're probably not going to use, and fluorophores and what we need to know about them. So let's get the ball rolling here. Spectral distribution. In the first initial introduction to fluorescence, I said that we have different fluorophores. Here we have tryptophan. So I'm just going to write here, this is tryptophan. Can get excited, these fluorophores can get excited in a specific range of light, and they can emit a specific range of light. And this is called spectral distribution. And here we have the spectral distributions for tryptophan here. So let's see what we have here. We mentioned that for the excitation or absorption, we have specific wavelengths that we can use for tryptophan. And this is anything that's under this blue curve here. All these wavelengths from this range to this range here can be used to excite tryptophan. This also means that anything in a longer wavelength or in a shorter wavelength cannot be used. Tryptophan would not be able to take that photon energy and use it to emit a photon to fluoresce. Accordingly, this is the, and actually I'm going to label this here, it's really important, excitation and absorption. These two are interchangeable. So it's really important to understand both and to know both. Excitation and absorption, this guy right here. And for the group of wavelengths, the spectrum of wavelengths it can emit, we have the emission spectrum, which is these wavelengths here, roughly from 450 to 600. Which means that the photons that tryptophan will emit would be in this range right here. Which also means that we can't expect lower wavelengths or shorter wavelengths to be emitted in a fluorescence process by tryptophan. I'm going to write it here. This is the emission spectrum. Perfect. And something that is very obvious to us right away is that the absorption or excitation spectrum, this spectrum right here, is being shifted to the right side in this graph, or shifted towards the longer wavelengths or the redder colors. And this is called the Stokes shift. It's important to know the Stokes shift. The shift from the excitation spectrum shifts towards the longer, the redder wavelengths, and that's why it's also called a red shift. A red shift. Now it's important to understand that this scale here, the x-axis, is basically the wavelength. This is the wavelength. So it corresponds also to the frequency, which means that here I have higher frequency and here I have a lower frequency. And it also means that I'm losing frequency in this shift. And because frequency is essentially energy, I'm losing some of the energy in this shift. And we need to account for that energy, so let's try and understand where that energy goes and why we have that Stokes shift. And I'm really quickly going to draw the Jablonski diagram, not all of it obviously, just the different levels. Perfect. And here I have my intermediate levels, etc. And I said that my electron in my tryptophan fluorophore can be excited and will jump up here. And if you're not totally familiar with the Jablonski diagram, I urge you to go through that video about the Jablonski diagram to get an understanding of it. But once it is excited, following Cauchy's rule, it's going to spontaneously and really rapidly vibrate down to the S1 level here in internal conversion. And this is a non-radiative relaxation, which means I'm not getting a photon from this deal here, from the internal conversion. And after that, we may emit a photon as well, which is cool. And if I look at these two levels, this is the energy that I was excited with, the tryptophan was excited with, and my photon emitting potential diminished because of my internal conversion here. And this internal conversion, this loss of energy, is why we have a shift here. And you can imagine that this internal conversion can take place from any energy level here down to here, and I may lose any range here of energy. So essentially the internal conversion, following Cauchy's rule, the spontaneous rapid non-radiative decay or relaxation to the S1 stable state, S1 semi-stable excited state, is where some of the energy is going, and that's why my emitted photons are going to be red shifted. And it's important to understand, oftentimes students actually know to explain one term and the other as well, but sometimes making the connection is quite important, making the connection between these two processes, because I find it helpful to remember what is going on. And that's why when you see a different spectral distribution, you'll see the excitation, and then you will see the emission, and it's going to be redshifted, etc., etc. And before we finish off with this graph, being that it's a graph, I'd like to go through the y-axis here, which is the intensity. And the intensity basically means that I have a peak here in my emission spectrum, which means my intensity peaks here, which means that if I go down, this wavelength here is going to be the wavelength I will see most photons in. I will see most photons in this specific wavelength when tryptophan emits a photon via fluorescence. This is the emission spectra maxima. And I also have the excitation spectra maxima, which is, this is the wavelength right here, which is ideal to excite this specific fluorophore. So this is the excitation maxima or the emission maxima. So this is our spectral distribution. And being that we have, in the presentation, we have, after the spectral distribution, this little depiction here, which often is asked about, I'll go through it. It's called, this little experiment would basically help me determine these spectral distributions, which means this will help me determine what wavelengths I can use to excite a molecule that fluoresces and what wavelengths I will get emitted. And this is called Spectrofluorometer. Spectrofluorometer. Basically what happens is really, really simple. I have polychromatic light. Polychromatic light is just light that contains all wavelengths emitting from here. I split it up, and then I can just pick one region and shine it on my sample and see if that sample fluoresces. If it doesn't, I can try this segment and see if it fluoresces. And by law, by elimination, I can actually determine which wavelengths I can use to excite my sample. And after I know that, I can take all the possible photons that my sample emitted, all the different wavelengths, I can split them up and I can detect them. And that way I can determine both of my spectrums. And this part right here, when I limit my emission, my excitation wavelengths, I can determine my excitation spectra. And at this point, I can actually detect what photons were emitted, and I'm just drawing this arbitrarily, I can determine my emission spectra. This is a spectrofluorometer. If you ask me if I've ever seen this represented in an exam, I will say no. But it's good to know because it's there and it's often asked about. Perfect. So now what we know of that, let's go through fluorescence parameters. And when we say fluorescence parameters, it's really a way to characterize different fluorescent molecules. We're going to mention quantum yield, and this is a pretty important way to characterize a fluorophore. Let's just say this is my fluorophore. Let's stick with tryptophan. Why not? And let's just say I'm sending 1, 2, 3, 4, 5 photons to excite tryptophan. And I'm asking, how many photons is tryptophan going to emit? Obviously, it can emit more than it absorbed, but let's just say it emits 3. So it emitted 3 photons, and I used 5 photons to excite it. So the quantum yield based on this observation here that I made for tryptophan is 3 to 5. And this is what is meant here. And what's meant here, because this is also obviously asked about, number of photons emitted are the total amount of photons that fluoresced, this is F, and these are the total amount of photons, the photons that fluoresced, the photons that went through internal conversions, the one that went through intersystem crossing, and you can really think about the denominator, this is the KF, the total amount of photons emitted. You can think about the denominator as the total amount of photons emitted plus the total amount of photons that were excited in a non-radiated form. And this is basically internal conversion and intersystem crossing. This is what is meant here. Another very important fluorescent parameter is fluorescence lifetime. And let's read it out. This is from the lecture slide, obviously. Fluorescence lifetime is the characteristic time that the fluorophore spends in the excited state. And what does that mean? Real quickly, we're just going to understand it with the different energy levels. This is my ground state. This is my first excited state. This is my second excited state. These are all the levels in between. So what I have here is an electron that got excited in my fluorophore molecule, got here, went through intersystem internal conversion, it vibrated or rotated down, and then it emitted a photon and fluoresced. And when you think about it, before it emitted a photon, from the point it was excited, which is this point, up to the point it emitted a photon, which is this point, this is the lifetime that fluorophore spent in the excited state. And this is the lifetime that we mean, this lifetime here. This is the lifetime that we spent in the excited state. And this is what I encountered, this is the definition I encountered in any, this is the definition here that I encountered in any article I read about fluorescence. But we also need to know that our department formulated minimal 104 in regards to that. Let's go through that, minimal 104 here. What is the definition of fluorescence lifetime? It is the time during which the number of excited molecules decreases to 1/8 times, which means 37% its initial value. And this is a different definition. So we need to understand that as well. What I'm going to draw here is going to draw, let's say, a little tank. Well, you can think of it as any way you want. And what I have here, say I have here fluorophores. Perfect. Now what I do, and this is the same tank. What I do is I excite these fluorophores with light that they can absorb. These are all the same fluorophores. I shoot them with light and they're all excited. So we have here at this point in time, this is, let's say, time 1. At this point in time, we have 100% of my fluorophores are excited. And slowly after a while, let's say this is the end point, Tn, the end point, all of them relaxed in a radiative or non-radiative form, and 0% of them are excited. They're back to where they were. Somewhere in the middle, before it diminishes to 0, this point here, it diminishes to 37%. 37% of my molecules are excited, and this is what they mean the time of fluorescence lifetime. This is what they mean by minimal 103, the time during which the number of excited molecules decrease to 37%. So effectively, it's the time between this time and this time. And although it's different from anything that I have seen in different articles, and also different from what I've seen both in the book and in the lecture slides here, the other way in the minimal to describe it is also a viable physical quantitative way to define a lifetime. So you need to know both. What I would recommend is if you're answering what is fluorescence lifetime in a minimal portion, of an exam, I would give the minimal answer. But both should be acceptable, but obviously I can't sign on it. So let's move on and talk about what's going on here. During this time, in the excited state, the fluorophore undergoes multiple interactions with the environment. So what we need to know is that when a molecule is in its ground state, it's relaxed. When it's in its excited state, just like here at this point, when it's in its excited state, it's subjective to different forces. It's subjective to rotational motion, to intersystem crossing, to fret. We're going to go through fret later. And it may collide with its environment until it loses its energy, collisional quenching. So these are the other forces that this molecule may interact with. A little bit about what's going on here, and before I read this out, this whole thing, before I read it out, I'm going to explain it because that's the way I'd like to do it. I'm going to draw some more containers here. Some more containers. And in these containers, I'm going to put, in this top container, I'm going to put a solution. And in this solution, there are fluorophores. And this solution is homogeneous. This solution is homogeneous, which means that it's uniform throughout. And the second type is going to be just a mixture, a heterogeneous mixture. And a heterogeneous mixture, as you can see, is not really uniform throughout. Perfect. So what I'm going to do here is I'm going to shine excitation light on this container here, and then I'm going through different time points, time point 1, time point 2, measure the lifetime I'm getting in this homogeneous solution. And because it is uniform throughout, the interactions should be ideally pretty much the same at any given time point that I measure. On contrary, if I have a heterogeneous mixture, over time, let's just say this is time point 1, and at time point 2, the system may look a little different. Let's just say that the system, because it's a heterogeneous mixture, things move around, and you can expect the parameter that I am looking at, the parameter I'm checking, to be different at these two different timelines. And if we read off of the lecture slide here, homogeneous system, fluorophore and uniform solvent, should in principle exhibit a single lifetime, which means that we'll see a uniform parameter. Or a heterogeneous system, most real systems such as cells typically show multiple lifetimes, which means that if I'm looking at lifetimes at different stretches of time, I may measure the parameter, I may get a different value for that parameter. And we've explained it here with the different mixtures. Perfect. So now that we discussed the fluorescent parameter, the major fluorescent parameter, all we have really left is to look at fluorophores. And obviously, by now, you should know that fluorophore is any molecule that fluoresces. And we have a lot of fluorophores. And those are basically in two categories. Fluorophores are in two categories. The exogenous fluorophores, this is an E, and the endogenous fluorophores. And exogenous, I believe, means outside or out of a system, and endogenous, I think, means within and system. Basically, this means that exogenous, out of the system, are they're not naturally occurring, which means that we don't see them in our bodies. They're not naturally occurring in, let's say, biological system. And these, the endogenous fluorophores, are fluorophores that exist in our body. They are naturally occurring in biological systems, occurring in biosystems. As I'm making this YouTube video, I have endogenous fluorophores in my body. Exogenous fluorophore, an example could be any fluorescent dye, like let's say fluorescein, C. And an endogenous fluorophore could be, let's say, the three amino acids, and they're in the minimal, so I'm going to mention them. tryptophan, tyrosine, and phenylalanine. And you don't really need to know all the name. You just need the three-letter aberration is enough. There are obviously a lot more molecules that fluoresce that we'll learn about, but there's no real reason to mention them. And the reason that I'm mentioning this specific information is because, first of all these three are in the minimals. And also, the way I prepare myself for an exam is if they ask me about fluorophores and they ask me to give an example, I can give any example. I can give fluorescein, I can give these guys. If they ask me about what are endogenous fluorophores, the ones that exist within biological systems, these are good examples to know. So whenever they ask about defining something, give an example, it's always good to know one example. Obviously, the lecture slides has a lot of examples, but I make a point to only know one of each category, just for sakes of exams. So hopefully you found this helpful. I'll see you on the next video.